Good evening, everybody. We're incredibly lucky to have Lindsay Marshall speaking with us this evening. Dr. Lindsay Marshall completed her bachelor's degree in biological sciences at Stirling University, followed by a master's of science degree in cellular and molecular pathology at Dundee University. She remained at Dundee for the PhD, where she studied the biology of the human airways with a particular focus on the disease-related pathology observed in cystic fibrosis and lung disease. She spent several years carrying out research into the defence and repair responses in the human airways and was senior lecturer in immunology at Aston University, where she used human cells to develop multi-cellular models of the upper airways. Her research team used these human-relevant models to investigate disease processes, the response to bacterial infections, the effect of cigarette smoke and e-cig vapours, and to test potential therapies. In 2016, Lindsay joined Humane World for Animals, where she now serves as Principal for Science in Animal Research Issues. In this role, her overarching goal is to accelerate animal replacement across all areas of research and testing with a focus on the United States. She works closely with non-animal method developers and policymakers and maintains a strong interest in identifying and advancing non-animal alternatives to animal models for investigating human disease mechanisms and treatments and for regulatory decision making. That's quite a lot.
Yes, yeah, thank you, Alan. That's nice of you to go through it all. I appreciate that.
Very lovely to have you. If you want to take it away, thank you very much, Lindsay.
Okay. All right, thank you. So thank you so much for the introduction. Thank you for the invitation. It's really lovely to be with you this evening. And luckily, Alan's covered most of what's on that first slide, apart from Franny. So this is Franny, who, can you see my pointer if I move my pointer? Can you see anything moving? Can you see my cursor at all?
No.
No. Okay. If I do now, can you see Elizabeth? Yeah. Can you see? Okay. So this, I didn't really need to point at her because you could see her quite well. This is Franny, who was one of the Envigo beagles. So you might have heard of the Envigo case in the US, where it was Peter actually that started the investigation that showed that these were dogs kept in a breeding facility in just absolutely disgusting conditions. And the Department of Justice in the United States asked Humane World for Animals to help them with the rescue because they didn't have the capacity to get thousands of dogs out of those conditions. So Franny was one of the dogs rescued. And as you might imagine, quite a few of those beagles ended up going home with Humane World for Animals staff because as soon as they were part of the rescue and they met the dogs and they met the puppies, those dogs got siphoned out pretty quickly to be home there. So she was destined for a life of research, but thankfully for her, she didn't get into it. And my disclaimer, I guess, would be that any opinions that I state in this talk are mine and mine alone, and they're nothing to do with my organisation. So what I'd like to talk about today then is a little bit of background about where are animals being used and how many animals are being used. And I appreciate that for my audience, you're probably all very well aware of this, but I thought that it's maybe good to have a reminder of what we know and what we don't know. And certainly as we come up to World Day for Animals in Laboratories, we can kind of think about what's happening to those animals a little bit and how many it impacts. So I'm sorry if this is kind of duplicating evidence that you already have, but it's kind of helpful for me to help me sort of set the stage as to why this science is so important. So then what I'd like to go into is really what do we need to think about for full replacement of animals and get into a few case studies of where we've got these really fantastic methods that are showing us exactly why we don't need animals and why this human relevant science is better. And then a little bit really at the end for us to kind of think about what more is needed. And I am a scientist, so it's going to be from a very kind of sciencey perspective. I will try to reduce my jargon, but please throw questions at me at the end and we can go over this and I'm more than happy to clarify or, respond to anything that you need me to. So if we look at where are animals used for scientific purposes, ironically, we're still in the dark, even though the bits on my map that are white are where we don't really know what's happening to animals. We don't really know how many animals are being used. We don't really know where they're being used. So it's very difficult for us to work out where do we need that scientific intervention if we don't yet know what's happening to the animals. So that's kind of my point about this here is that where we've got light blue areas on this map, there are some records. And by some records, I mean there might be some areas report animal use. There might be some species are reported. There might be some purpose of use reported. And by reported, what I mean here is publicly available. So this is not data that we're having to dig for with freedom of information requests. This is data that's already out there for us to be able to see quite easily. the UK and the European Union are in the darkest blue because that's where we have the most detailed records. So we have purpose of use and species and severity and number. And that's helpful, not least because it helps us realise there's still a lot for us to do. And that's it's kind of sad, but I think it's good to remind ourselves that this is the extent of what we're dealing with. But also, I think from my perspective, from the work that I'm trying to do, so from my professional life, it's useful to have that granularity of data so that we know where we have had an impact and where we need to keep working to have more impact. So given that I work in the US, I'm not sure how many of you will be aware that for the United States, the only animals that they're legally mandated to report use of are kind of the biggest species. So I guess, sorry, to flip that round. In the United States, there are no requirements to report the number of mice, rats, fish, or birds who are bred and used for research. So if we look at the numbers in the US, they make absolutely no sense. So the actual recorded use of animals in the United States suggests that it's around about 750,000 animals are used a year, which is crazy, isn't it? Because we know how many are used in the European Union and in the United Kingdom. Now, what's kind of interesting is there's been some, I suppose, pushback on this lack of transparency from one university at least. So the University of Washington in Seattle has decided that they're going to be transparent. And the links in the papers that I've got on these slides, I'm more than happy to share them afterwards if that's helpful for you. So I know you can't write all of that down anyway, so don't worry, I'm happy to share my slides. So what the University of Washington have done has actually report on the number of animals that they record officially. So that's what's required, that's publicly available, which is 13,306. And then the number of animals that they actually used, which is 338,437. So that's when they include the mice and the rats. So we see that when we look at the data from the United States, we need to be aware that it's vastly underreporting the number of animals that are being used. And I did a quick kind of, like back of a bus ticket calculation and sort of and said that, okay, if we look at the University of Washington in Seattle and say that it's using this 338,000 animals, and we estimate that around about 187 universities in the US are about the same level of kind of research intensity as this university, then we start to get a better picture, I think, of what the extent of animal use is in the US. And that gives us a figure of 63 million animals, which is probably closer to what we might expect if we think about the EU animal use. But that's just for US universities. That's not thinking about industry. That's not thinking about pharma. That's not thinking about the contract research organisations. So I think we're really looking at the tip of the iceberg when we're looking at these publicly reported figures from the United States. I think it's hard to be kind of proud of animal use, isn't it? But I think at least in the in the European Union, we have this database that's really great for us to use. So I don't know how many of you are aware of the Allures database, whether or not you've ever used this database. I use it a lot because it gives us a real granular picture of what's happening to the animals. And it parses it out to these three separate sections. So section 1 is the actual numbers of animals. Section 2 is the purpose of use of animals, which is one that I use the most often. And then section 3 is focused on genetically altered animals. So I thought, if it's all right, I've just got a couple of slides that look at how we might track through this Allures database to find information it's helpful for us to see what's going on. So if we go into section 2, you'll see that you get these different filters. So I don't know how clear that is for you, whether you can see it, but the only one that you're required to insert is the reporting year, and then it will give you all that data. But what you can do is also tell it that you want to see how many of those animal uses were reuse of animals. So this is, again, the purpose of use, but this allows us to see whether or not that animal has been reused, basically. So we can have a look at that. And we know that reuse is kind of popular as a refinement because it makes the numbers look lower. But I think we have to consider the lifetime experience of the animal who is being reused. And actually, I'm not sure that whether I'm really such a great proponent of reuse, because it depends, doesn't it, what's happening. I know there are limitations on what can happen to those animals before they're reused, but it still doesn't get us closer to that improving the science or the methodology to get that animal out of the laboratory by having this reuse. And currently, I think reuse is around, it's certainly less than 10% of all the animals. So I think that's an indicator as to what's happening to the animals, the severity of what's happening to them, the fact that they can't be reused, and that we know that a lot of these procedures are terminal, certainly for regulatory purposes. So I've kind of pulled out dogs here just to have a look at what we can get from Allures in terms of what's happening to dogs. That's not because I think that dogs are any more important than the mice and the rats and the birds and the fish and everybody else. It's just because the numbers are a bit easier to go through. And one of the projects that we're working on is looking at the use of dogs for pesticide testing. So I was kind of taking those data for that as well. So if we look at the high level purpose of use of dogs, we see that basic research, which is kind of that research which is just in the pursuit of knowledge, so it's not really linked to any kind of disease or drug development or anything like that, that's around 1,300 dogs. Translational and applied research, which might be where a dog has been given a disease or a condition, or we're trying to look at what an animal disease might progress like, is around 5,000 dogs. And then the majority of use of dogs is for regulatory use and routine production. So that is testing, basically, so the toxicity testing and testing that is required at some level to be able to get a product approved. And we can dig into this a bit more in a bit more detail. And what we find is that this regulatory use is for legislation on medicinal products for human use. And most of that is for toxicity testing. And I just think, I think we kind of need to look at that for a moment and just think about how ridiculous it seems, frankly, that this is 4,500 uses of dogs for legislation on medicinal products for human use. And yet I imagine that everyone on this call understands the differences between dog metabolism and human metabolism. And we know that, you know, the signs we get at Christmas time about not letting our dogs eat raisins or chocolates. And yet there are thousands of dogs being used to satisfy legislation for medicinal products on human use. And I thought, as I kind of move into the sciencey side a bit more, this might be helpful for us to think about what drug development sort of currently looks like and the extent of animal use across this pipeline of drug development. And we start at the top with when we've got maybe thousands of chemical compounds that we're potentially interested in, that we think might be useful, but we're not really sure which one might be. And then you see this kind of filter effect as we move down towards selecting something that we might be helpful. And then on the right of the screen, you'll see all the different kinds of animals that are involved in this testing. And I just wanted to make the point here really that this drug development, as we know, is heavily reliant on animals, all these multiple species, rodents and non-rodents. Many of the use of these animals is recommended in guideline documents, so it's really difficult for pharmaceutical companies to make the argument to move away from it, even if the methodology was there and they wanted to. But what I found, I guess, an interesting observation I'd like to share with you is that I was at a workshop meeting with a bunch of pharmaceutical industry scientists, and one of them said that basically, irrespective of all this animal testing, at some point, our test compound has to go into a human. So it has to be, there is always going to be a first in human. The point of all these bodies of animals that pile up underneath that compound is to try and see whether or not it's safe. But the differences between animals and humans mean that it's still a big risk when we go into humans. So I think one of the changes that I would like to see is how we regulate voluntary use of humans in clinical trials and kind of, you know, early clinical testing, and how we make sure that that's safer for people to be part of, so that we're not as reliant on these animal data, and actually we can use these different methodologies that I'm going to talk about to get a compound that we've de-risked, so that we're fairly confident we can give it to a human and not have a disastrous effect, rather than all of the animals. And then I wanted to, I suppose, have a bit of a positivity. I feel like all those animal numbers are kind of a bit they're a bit gloomy. It's not something that we particularly want to think about too much. So I wanted to have a quick roundup of what's been going on, because there's a lot happening in the past year or so. There's been a lot happening globally, and a lot of that, we can make a very positive picture out of it. There's still a lot to be done, but I think we're seeing this real global intent to phase out animal testing. And I know we've been there before. We've heard this for a very long time, but I feel like as my work is US focused, this feels like the first time since that we've had these real federal agencies making these bold statements. So we've had various roadmaps. We've had the FDA is the drug regulatory agency in the US. It published its roadmap almost a year ago. The National Institutes of Health, so the NIH, is the world's largest funder of biomedical research. It has a budget of around $42 billion every year, of which about half goes onto animal research. And yet in 2025, June 2025, they announced that they would no longer fund any new applications that relied solely on animal use. So it's very nuanced. So it's only new applications, and it's only those that rely solely on animal use. But it's still the first time we've had something this bold from an agency that has this budget. So it feels like this is a move in the right direction. The NIH then announced also a couple of months later that they were developing, or they were initiating, a standardized organoid modeling center. So this is what we'd call purely human relevant science. This is the first of its kind. So they're developing this center that will, excuse me, that will be responsible for developing the methodology for people to be able to make human organoids. So this is models of human, I think they're starting with lung, liver, kidney. And it's not about the distribution of the organoids, it's really about developing the methods so that anybody in the US can get this standardized method and develop it in their lab and use it in their lab. So that's, if we're thinking about everything that we need to be able to change the system, we need the money. And the NIH have said, okay, they're moving in the right direction in terms of where they want the funding applications to come from. And we need the sort of training and education. And that's where I see this standardized organoid modeling center is step one in developing training and educational tools for people to be able to make that move. We then saw the UK government publishing its roadmap and saying about its 75 million funding, which it kind of had already given out. So I don't really feel that's not as positive, maybe. We had the Environmental Protection Agency in the United States recommitting to ending all testing on mammals by 2035. And we kind of have to celebrate it a little bit, I think, because it's a move in the right direction. But honestly, it's a little bit disappointing that it's mammalian testing when this is an environmental protection agency that isn't considering fish. So it wouldn't even, make that move towards vertebrates. So I think there's a bit of a big gap there. And okay, maybe that'll be feasible to do by 2035, but it's hardly ambitious, and it's hardly getting us to the point of phasing out animal use for ecotoxicity testing. In the UK, I think last month, it's kind of exciting to think that we've got these invitations for this preclinical research hub that was in the UK roadmap. So at least step one has started there, that the MRC have asked for people to apply for that. I think that's one for us to look out for, really, to see who's going to get it. Is it going to be Oxford with their massive animal facility, or is it going to be somebody that we maybe, I don't know, has clearer intentions towards replacement rather than reduction and refinement as well? Last week, the US FDA released its NAMS guidance for industry. So this is, by NAMS, I mean the non-animal methods, or the new approach methodologies, these kind of animal sparing methods, which is frankly underwhelming and puts a lot of pressure on industry to provide the evidence that FDA need to see that they will then allow acceptance of those data. So it really feels a bit like, I don't know what the right word is really, but I think it's the FDA fulfilling something in its roadmap without actually moving things further, if that's fair. And that's very much my personal opinion. And then we've seen the NIH actually commit $150 million to technology development centres, which are a bit more promising, because this is looking at the kind of organ chip models, these really advanced, sophisticated non-animal models that cover a breadth of projects, actually. So they've got a project looking at women's diseases, they've got a project looking at chemical screening, they've got a project looking at neurodevelopment. So these are areas that are really important, that we start to develop the methodology that hopefully can then cascade beyond that. And then I think probably one for all of our diaries is that European Commission's roadmap is due potentially next week. I think they've said by the end of March. So hopefully that will be hugely ambitious. I really hope it's going to be hugely ambitious. I feel like they've had the stakeholder input to allow that to be a little bit more ambitious. So we'll see what that looks like. So thinking about all of this then, how can we really replace and reduce animals? And I've got reduce here because I think it's the step that I'd rather talk about. I feel like the 3 R's, we know the 3 R's have been around for longer than me, which it feels like not a lot of things have, and they haven't really aged particularly well. But from a professional perspective, we still use the 3 R's because it's embedded in legislation and it's the language that the industry use. So I think it's still in my vocabulary as much as I'd rather it wasn't. So I think in terms of the 3 R's, I only really want to consider reduction and replacement. And reduction is possible in terms of testing on animals for regulatory purposes, because we've seen agencies allow waivers. So, reducing animal use, not having to use animal use where there are other data that exist, use of existing animal data, the reuse that we've already kind of touched on, possibly virtual controls. And I suppose they add up to something, but what we really want is to think about the replacement, not just for the animals, but actually because these novel methodologies, if they're built with human cells and using human data and really entrenched in human biology, then they're better at predicting what's going to happen to humans, what's going to happen to patients. So I don't need to argue to you why we need to go to replacement, but that's kind of, where I'm sort of coming from, that reduction plays a role, but only if it's reduction with a view to replacement. We're not happy with just, we don't really want to just stop at waivers. We want to make sure that those waivers are going to lead to a real clear strategy for how do we get to the next step. And in order to do that, I think we do need a new approach. I think we really do need to step back and think about what are we asking for. I don't know how many of you will know of Thomas Hartung, who used to work at the European Centre for Alternatives to Animal Testing and now works at Johns Hopkins University in the United States, and is a toxicologist, an expert in computational biology and kind of neurodevelopment. And he was the first person that I heard say that people are not 70-kilogram rodents. It's absolutely true. We know it's absolutely true, but people are not 70-kilogram rabbits or dogs or monkeys either. So I think we kind of need to expand that and just think about, if animals are not predicting in what happens in people, why are we still using these animals? And I know that you all know all of this already, that we know that at least 90% of drugs fail, even though they appeared safe in animal testing. And that percentage goes up to 99.6 if you look at Alzheimer's disease. And I think that reflects the complexity of these neurological conditions and the fact that quite often we don't really understand what's happening in people, and yet we're attempting to model it in animals and then try and get those animals to tell us what drug might be helpful. And we have no idea what's going on with the human disease. And so that failure of the animal model is associated with the insurmountable species difference that we see between any other non-human mammal and a human. And on a kind of more positive note, we're seeing investment and engagement with these non-animal methods. So although the market for animal methods is still relatively high in terms of value, it's still 10 billion, or it was valued at 10 billion in 2019. You'll see that these compound annual growth rate for the non-animal methods is starting to overtake the compound annual growth rate for animals. I'm not an economics expert. I'm just kind of thinking that if this number is bigger, that means that this line is going to be steeper for the NAM. So we're going to have more investment, more interest, more uptake of these non-animal methods, so that, the tide is turning. Not quickly enough, but it's turning. And this was a publication that Katie Taylor did, that you might know of Katie Taylor. She used to work for Cruelty Free International, but I think she's freelancing consultant now. So she looked at the sort of more sciencey picture. So we've looked at the economic investment. This is kind of looking at the academic interest in the non-animal methods and looking at the number of papers that have been published, either using animals, which is this dotted line, or using the non-animal methods, which is this top line. So it's a concern that the number of papers using animals is still kind of ticking up. You know, we're told all the time, aren't we, about how animal use is reducing, and look at this, and we're using fewer, and it's so much better, and it's a positive picture. That kind of bothers me, that there's still that academic interest in the use of animals. There are still, frankly speaking, people building their careers on the use of animals because they still can. But I think what we can focus on is this, the fact that now the non-animal methods are overtaking animals. So hopefully this is an indicator of the education and training starting to shift now, so that we've got young scientists coming in who are now able to use the non-animal methods throughout their career, don't have to learn on animals and shift, but can actually have a completely, effectively cruelty-free career, I think. So what we want to do then is take our research and testing toolbox from him to, this is an organoid. This is a stained picture of an organoid. So this is a miniature organ model. These are organs on chips. This is a 3D bioprinted organ. And this is kind of my kind of estimation of what computational modeling would look like. And these, I would suggest, are all kind of human biology relevant. This is human cells, human cells, human cells, human data. And I'm not really a massive fan of kind of the artificial intelligence and the computers and all the rest of it. But I did find a slide from a talk from somebody from CN Bio, which is kind of a non-animal method developer in the UK. And it did kind of make sense, I think, that we've got artificial intelligence now, so we should be able to use it more effectively. And I think for us, or for our community, more effectively means, how do we replace animals using artificial intelligence? And this just really indicates the how these non-animal methods, including artificial intelligence, can be used across the breadth of that drug development pipeline and replacing animals at every single stage. So this, to start with, this target identification and validation, that would have been mice and rats. So that would have just been seeing whether, seeing whether or not something was expressed in a mouse or a rat. Could we hit it with a compound? What happened? Now, maybe we can use data from previous experiments, or from what we know about the human genome, or for what we know about human cells, and start to do that target identification and validation. And the same at every step, really. I think we can start to harness the data that we already have to reduce animal use, and ultimately to replace it by building these better artificial intelligence kind of based models. The important thing about this is that we need the data. So it's a call that, like, my organization has, is that we need more transparency. We need data to be shared more openly, and we need data to be shared in a way that's machine readable. It's not enough for pharmaceutical companies to say, we're sharing our data, we're publishing the papers, we're releasing our clinical trial data. That's not really helpful. That's not going to help us build quality machine learning models that can mine those data and find the information that we need, so we're not duplicating research and we're not using more animals when we don't have to. So that's my plea for more data. And then now I've got a couple of examples that hopefully will work. Okay, yes, is it working? Can you see things happening? Brilliant. So this is a blinking eye on a chip model. Now, it's not supposed to recreate an eye, so it's not meant to look like an eye. This is really meant to recreate the function of the eye, and not in terms of vision, but just in terms of, this is looking at the cells that are on the surface of your eye, basically. And can I play it again? I probably can't play it again. That's going to be beyond my capabilities. Sorry. It did work before when I did this. Is it because I got this? If I do... No. Sorry, I need my 19-year-old son to come in and help me. The point of the blinking eye in the chip was that we could start to look at what's the impact of diseases like dry eye disease, diseases that might impact the release of tears across the surface of the eye. What's the impact of different chemicals if we put them on the surface of kind of a human eye? And we can use these human cells with that kind of strangely blinking eyelid to really recreate what happens in a more human eye without using rabbits and without using monkeys. So that's, it's a very, I guess, a niche example, but it still shows the complexity of model that we can build in the lab, that we don't need to have an animal to blink its eye, but we can have that mechanical setup, which I think is quite beautiful. And then I've got another example. If I can get my pointer to work now. No, my mouse isn't working. Okay. Right. Now my mouse is working. Okay, sorry. So this is close to my heart, the breathing lung on a chip. So before I click play, actually, just to explain it very briefly, what we have is the grey is the membrane in the middle, and we have our lung cells on the top, and they're sitting in the air, which is where lung cells like to be. So they're happy there. And we have endothelial cells in the bottom, and they're in liquid, and they're like, imagine they're like your blood vessel cells, so they're much happier in liquid. So the advantages to this sort of setup are that, number one, we're using human cells. We can increase the complexity of this as much as we want. So we could have different cell types on here to build up different cell types that we'd find in the lung. And we've got really easy access to this channel underneath the cells and this channel on top of the cells. So that's really important, that this is, broadly speaking, not possible in animals. So it gives us an advantage over animal models, not just that it's human, but it's easier for us to analyze what's going on. And what this breathing lung on a chip does, the most important part about this is that we can put a kind of this vacuum across the membrane to stretch the membrane and make the cells expand and contract as if they were in a breathing lung. So exactly as if you were breathing in and out, we can recreate that physiological mechanism. And when they do that, what they found, these researchers found, is that you'll get the same response that you get to certain drugs in the clinic. So this chip was set up to try and investigate why people who are on chemotherapy and taking this compound IL-2 end up with fluid in their lungs. And if I can get this to work, oh, it's not going to work now. That's very disappointing. Oh, now it's working. Okay, so you'll see you have the blood flow underneath and nothing happens in this sort of static culture. But when you start to stretch it to make it think like it's bleeding, then the blue on the top there indicates that you've got fluid moving into that upper channel. So as someone who's interested in lungs, that's really exciting and thrilling and wonderful. But as people who are maybe not so excited in lungs, what this really showed us was that we can recapitulate what happens in an entire human using this lung chip. So we could show that this compound was creating this pulmonary edema, so this fluid exudation into the lung, using this tiny model that's about the size of your thumb. And that's important from a biological perspective, that we can show that this recreates what's happening in a person, but it's also really important from a drug development perspective, that we can start to look at how do we stop this. You know, we don't really want to just look at these lungs filling up with fluid and say, oh, wow, that's amazing. We want to work out what that mechanism is so that we can stop it happening. And the lung chip allows us to do that. Okay, so we don't want to go. Okay, so this is where I kind of see the future of these methods, I think. So this is the human on a chip. And obviously it doesn't kind of look like this, but really what we're thinking about is, how do we get something that's more complicated than just that lung on a chip? And really, I see this as a way to counter the argument that people use animals have, where they say, we need a systemic system. We've got to have a whole body system. We need to still use mice because we need to see what happens in the whole body. We can't do that with these non-animal methods. actually, I think you can, or this is the way that we can start to think about doing that. Okay. We don't need a whole body. We just really need to think about what's those important organs that we're interested in. And that's where this human on a chip allows us to look at those different organs. How do they talk to each other? How do they influence each other? We could use this to look at basic biology. We could use it to look at drug development. We could use it to see where drugs go in the body. We could use it for chemical screening. We can use it for everything that Allure's database says animals are used for. I think we can start to think about using this human on a chip. And the beauty of it, I guess, is that it allows us to ask questions of a human system, not an animal system. And also it allows us to really tailor what questions we're kind of asking. So if we're interested in an inhaled drug, then we put a lung on our chip and we have a look to see how does the lung respond to that. If we're not really bothered and something's never going to get to the lung, then we can take that out and we can maybe just look at intestines and kidneys. So we can make our experiments a lot cleaner than using animals. And I say that, I hope you understand the perspective I'm coming from here, that what I mean is giving an animal a drug is it's been likened to just a black box effect. So you give the animal the drug and then you kind of step back and see what happens, but you don't really know what's going on in anything inside that animal. This human on a chip allows you to define where you put the drug and then take precise measurements from all the different organs that you're interested in to see exactly what's going on. So there are advantages way beyond just it's not an animal, it's a human. I think there are real scientific advantages to this. And I guess a more practical one is that, so the human on the chip on a microscope slide, kind of the size of my thumb, requires a lot less experimental compound than any sort of animal, than a monkey or a dog or, you know, a pig or a mouse. So it's conservative in that regard as well. That's the least of our worries, but it's still, if we're thinking about why might pharma need to switch to this, I think that's something to consider, that it's going to be economically advantageous to them. So that's the human on the chip. And then the other thing that we can create with a similar sort of setup is the patient on a chip. So what this really means is a kind of disease model, but a disease model, again, that's very human relevant and allows us to retain all the genetic characteristics of that individual patient. So you can imagine that for some of these areas that we're talking about, it might be quite difficult to get access to them on a human. So people aren't understandably reluctant to give you samples of their brain if they don't need to. But what we can do is exploit the wonderful kind of flexibility of cells. So you can take a skin cell sample from people, and in the lab, you can persuade that to turn into any one of these cells on this patient chip. But you imagine if you've got, for example, the skin cells of somebody with Alzheimer's disease, you can get those cells to turn into brain cells or kidney cells or liver cells or lung cells, but they all retain the genetic signature of that Alzheimer's disease. So we know that we then have this real reliable model of our patient. And we can start now to have a look at the efficacy of different drugs for these different conditions. And we can use this to, you've probably heard of personalized medicine. So we could develop these, you know, individual patients on chips to look at what medicines would work for individual people. We can look at this kind of patient stratification to see which patients might respond to different medicines. We can use it for drug discovery and basic research. And this, maybe not an entire patient on a chip, but certainly I feel like this is starting to have traction, or this ability to use, I'm going to say bits of a person, but that's not right. The ability to use, for example, tumor samples. So there's really good examples of where maybe 10 years ago, people would have taken a sample of a tumor, put it into a mouse to create a xenograft model, and then use that to try and look at different chemotherapies to try to titrate out which different drug combinations will be effective for that tumor. And then months later, go back to the patient and give them that drug. And what clinical trials are doing now is still take the tumor, but grow it in the lab, in a culture without a mouse, and then use that model to look at those different combinations of drugs to see what might work, and then get that back to the patient within weeks rather than months. So we're kind of cutting out the mouse now. We're seeing the sense in cutting out the mouse. And I'd like to think that that would expand and we can start to do more with these, you know, organ chip cultures. Okay. And this is one area where I think we'll get really good traction with the organ chips, because animals are not giving us anything for this drug-induced liver injury, which quite often is idiosyncratic, so we don't really know why it happens, and we certainly aren't expecting it. Remember, I mentioned that we all know this figure of 90% failure rates for drugs that are developed in animals, and of that, around 30% is unexpected toxicity. And I couldn't really find how much of that is liver, but I think it's probably a great deal of it is liver, given that your liver is the organ that's trying to make these drugs safe, that, you know, that has that metabolic activity. And this drug-induced liver injury is a significant cause of acute liver failure. So it's important that we understand this, that we understand how it's happening, and we try and prevent it happening. So what we want to do is make sure that drugs are not getting into people if there's a chance that they're going to cause DILI. And that's where organ chips really have massive potential to help us out. So this is this image is actually, it's an organ chip made by Emulate, which is a company that spun out of Harvard University in the US, and are doing pretty well actually in terms of the range of organ chips that they offer, and the dedication they have to showing that these organ chips have the potential to replace animals. They're a pretty good company in that, I think, really. Well, certainly some of their employees are quite outspoken in the potential that they see for their chips to replace animals, which is always nice to hear. So they did the biggest study to date looking at these liver chips. So using, these are chips using human liver cells. And they took 27 drugs, which was a mixture of drugs that had been shown to cause liver toxicity in people and drugs that were shown to be safe in people. And remember that of these drugs, the animal testing had said all of them are fine. The animals had said that none of these drugs were hepatotoxicants. And yet we know that at least 20 of them were toxic to people. So the animals are saying it's all great, just carry on, give them to people. What this study found then was that when they tested these hepatotoxicants in the liver chip flagged 7 out of 8 of those toxic drugs as toxic. So it's 7 out of 8 times better at the animals, telling us what is toxic. And also, really importantly, the liver chip did not flag any of the non-toxic drugs as toxic. So it's kind of got that. It's able to show us which ones might be toxic. It's not perfect, but it's still way, way better than animals if it's got 7 out of 8. And the animals just said, no, they're all fine. And it really gives us, I think, well, what Emulate, the company, are saying is that we really should be using these liver chips quite early in drug screening to get that flag as to whether or not a drug is going to be toxic to the liver. And they're saying that if we use these liver chips, we will be able to reveal DILI, so the drug-induced liver injury, before we get anywhere near people. And this is really important because there are so many drugs that fail because of this liver toxicity. If it's tipping people over into acute liver failure, of course it's important that we weed out these drugs. And this is not necessarily to stop development of them, but it's to be able to modify them to reduce the drug-induced liver injury. And that's what the chip would be able to show. And an animal wouldn't be able to show you that because the animal is saying the drug is non-toxic anyway. So it doesn't matter how you modify a drug. If the animal says it's non-toxic, we can't reduce the toxicity of it and find that out with animal testing. But we could with the liver chip. And beyond that, in this study that Emulate did when they used those hundreds of liver chips, they worked out that the potential for increased productivity using a liver chip was $3 billion US. And then they said, if you added that to 3 additional organ chips, so I think they were talking about kidney, heart and lung probably, then you could increase the savings to $24 billion. So there's a great economic reason for shifting towards these. Now, I've got, if you're all still awake and still with me, I've got a few more slides of kind of case studies of where there's just real world examples of where the non-animal methods are being used by regulators and having real world success, I would say. And one of the main areas that we see increasing interest in the use of these non-animal methods is in rare diseases. And there are lots of reasons for that, some of which I've tried to kind of to map out on the slide. Practically speaking, we might not know what causes a rare disease, and that creates a problem for that traditional route of drug development, because normally I worked on cystic fibrosis for a long time, and as soon as the gene that is mutated in cystic fibrosis was identified, pretty much the first thing people did was put it in a mouse, make a mouse model, because that's how science worked, really. Find a gene, make an animal, get a drug. And now what I'd like to think we're starting to see that flip with these organ chip models, that now it's find a gene, let's look to see whether or not we've got a cell-based model that we can develop instead of using the animal. And even for not even find a gene, find a patient. We don't even need to know what the gene is now for these rare diseases to be able to find the patient, take some of the patient's cells, develop an organoid model or an organ chip model, and then let's have a look to see what drugs do. You know, so we can really completely cut out the animal and get that mechanistic insight into what's going on in that disease that we maybe know very little about. There may only be a handful of patients in our area, in our region, in our state, in our country that has this disease, which makes it really, really problematic to try and run a clinical trial. If we want a clinical trial with thousands of people, we might not have them, but we could try and do that on chips. So we could try and, if we know the gene, we could make versions of our patients and do it on chips. If we don't know the gene, we could try and, you know, use a different way to do it. But I think it's certainly more feasible than the old-fashioned mouse model way. So the one example that I've got here then is, this was one of the first ones that came out actually, and this is a really nice example of understanding what's going on in the disease and then really thinking about what drugs have we already got that might be useful in this particular disease. So these researchers took nerve cells. They developed these. They took stem cells and then induced those stem cells to create nerve cells. So, you know, stem cells are those multifunctional cells that we can make them do turn into different cell types. And then alongside the nerve cells, they got some primary Schwann cells, and the Schwann cells are the ones that create myelin that wraps around your nerve cells and protects them and allows nerve conduction to happen more effectively. But if there's damage to the myelin, then there's problems with nerve conduction. And the disease they were looking at, there's damage to the myelin. So they were creating this kind of healthy model with the nerve cell and the myelinating cells. And what they did was take serum from patients who had this condition and put it on their healthy model and showed that they got damage to the myelin under these conditions. So that's the mechanism of this disease. There's something in the patient's blood that's triggering that damage to the myelin that leads to the issues with the nerve conduction. So that gives us a really good clue as to what we might need to do to be able to block that. And the researchers found that it was a particular component in the patient's blood that they could block with this drug that already existed. So this is a drug that had been approved for a different purpose. So it means that it had already been through first in human. It was already on the market. There was less of a barrier for them to be able to use it. They showed in their chip that when they put the nerve cells and the myelin cells and the patient's serum and this drug all together, they stopped the damage from the patient's serum. Now, I mean, from a scientific perspective, I find that really exciting. But actually, from a regulatory perspective, from an evidence perspective, those experiments were enough for the regulator to say, okay, you can put it in clinical trial for that condition. Now, that's because it already had this approval for a different condition, but it's still, number one, it accelerates this drug getting to those patients who need it. And number two, it stops any additional animal testing, because at least the regulators are taking this efficacy data from this human method, and they're not requiring that it goes into an animal model of the disease. So I think that's something we can hopefully press for more of. And then this, sorry, I've gone back to lungs again. So this is another example of drug repurposing. And this was this is using a drug that had been approved for Alzheimer's disease. I'm going to say it's azelaregon. I have no idea how you pronounce that. These drug names are just ridiculous, aren't they? And I forgot to look up how you meant to say it, but it kind of doesn't matter. So this was... the mechanism of this drug was already known because it had been approved for Alzheimer's disease. So really what these researchers were doing were kind of looking at, well, okay, where might this biology have impact? And they thought, well, it might have impact in reducing infection. So it might be able to stop the entry of virus into lung cells. So they set up the lung chip. This is the same people that did that breathing lung chip. So they've got the background in it. They know what they're doing. And what they did was showed that when you had the lung chip with either influenza virus or COVID virus, and then they used the azelaregon, they could block the entry of the virus into the lung. And again, because this drug had already been approved for Alzheimer's disease, so the regulators were comfortable with the body of safety data that already existed, the drug was moved really rapidly into clinical trial. And it went to clinical trial in South Africa for COVID. Now, unfortunately, I think that kind of stopped because the COVID, the number of people presenting with COVID, thankfully, reduced, but it reduced such that this clinical trial couldn't really go ahead, or, it's still recruiting. They haven't got the right number of people. So we don't have data from this, but it's still another example of where we can really start to be clever, I guess, and think about the biology that we're trying to cure or treat, and look at the drugs that are already out there, and then use the chips to see whether or not this will be effective. So this is a really kind of effective and relatively quick way that we could start to screen lots of different drugs and work out what's going to work. And I think it's something crazy, like of the thousands of diseases that we've only got very few cures and only hundreds of treatments. So there's really a need, I think, to start looking at what do we already have and how might it be used more widely, or, in diseases that we maybe haven't quite thought of. And then this, I've only got two more, okay? So this is my favorite one. This is using a blood vessel chip. So this is using human cells again and creating a tiny tubular blood vessel, putting blood elements through it basically, and kind of really recreating your circulation effectively, but at a very, very small level. And this was set up to see whether or not this blood vessel chip could recapitulate what had happened in clinic. So we have the same old story. We have a drug that had been developed for an autoimmune disease that had passed all of its animal testing, that had gone through rodents and non-rodents, not a problem whatsoever. Got it to clinic, gave it to humans, and people started having quite problematic blood clots, which means that that drug has to be pulled from the clinic, obviously, because there's no way that we can give people something that's going to mean them throwing thromboses all over the place, basically. And the blood vessel chip actually recapitulated those blood clots, which I kind of find mind-blowing, really. So you've got your tiny blood vessel chip, you're flowing your blood components through it, you add this experimental drug, and they saw various indicators of blood clots forming in this chip. Okay, that's exciting, but it doesn't really mean a great deal for us as patients as we're thinking about drug development. The really exciting part about this project is that then the researchers took the drug, modified it slightly, kind of thought about its structure, thought about its activity. So they changed its structure, tested it again in the chip, and found that it didn't create the clotting anymore. So they managed to do a modification to alter the side effects of that drug that meant it had limited its kind of clinical life. And I find that really interesting because, again, that's something that we couldn't do in animals because this drug didn't cause blood clots in animals. It doesn't matter what modification you do. You're never going to see a reduction in blood clots in animals when they're not happening in the first place. But the chips then give us the capacity to de-risk drugs before they get to people, which is kind of important, or can we look at drugs that have failed in the clinic, take them back to chips, modify them, and see whether or not we can alter that so that we can improve or reduce the side effects of our drugs. I'd like to think that this is something that's going to be attractive to pharmaceutical companies if they think that they can use a kind of quick method to improve the efficacy, I guess, of their drugs, because that's going to improve their sales, isn't it? And that's kind of what they want. And then my final kind of example here is, it is a word of caution, I think. I'm going to end on the word of caution. I'm going to start with the really exciting stuff, because this was, the man in the picture here is Professor Muwatri, who works out of UCSD, so in California. And he's a fantastic scientist. He's done some absolutely brilliant work. He had a project called the Tooth Fairy Project, where he was inviting parents to send in their baby's teeth as they fell out. Obviously, the Tooth Fairy still, paid the children their money. That's, that was part of the deal. She was one of his collaborators, I'm sure. But he was extracting the stem cells from the pulp in these baby teeth and developing brain models, or like brain organoid models of these children. And his idea was to collect samples from kids who were healthy and kids who had neurodevelopmental delay or autistic spectrum disorder, and then use these models to look at the electrical activity in the brain and work out what are the differences between children that might have neurodevelopmental conditions and children who were apparently healthy, and then really think about, is there a way that we can modify that biology and improve the I guess, electrical conduction to try and not correct, but just to try and alter that neurodevelopmental delay if we could. So that was one of his projects. This is another one of his projects, was this Pitt-Hopkins syndrome. So they took stem cells here as well from 4 different patients with this Pitt-Hopkins syndrome and created organoid models with these patient cells. And they were the first, I think, to recognize the gene that was mutated in Pitt-Hopkins. So they did the comparison between the normal people and patients with Pitt-Hopkins disease to show that there was this gene mutation, and then developed a gene therapy to replace that mutated gene and use their organoid models to show that this gene therapy corrected the defect, I guess, in neural conduction or whatever it is. Sorry, I'm not an expert on Pitt-Hopkins syndrome, but it was the first example of this gene therapy being used in a lab-based system. And I think they went on to get to clinical trial with this. So that's why I took this from LinkedIn, actually. The screenshot is from LinkedIn, because it was very celebratory, which I think it should be. But then I thought I'll dig into this a bit further, because it seemed to me that people were saying that this has been a drug that's been developed. There's some element of artificial intelligence in here. There's definitely the use of organoids. It's definitely a human relevant system, and it's gone to clinical trial. And I thought, this is it. We've done it. This is exactly what we want. And then when you look a bit more closely, actually, the drug was also tested in mice and monkeys before it got to clinical trial. So you're kind of like, oh, we're back to where we were. So I think my note of caution, I think, here is just that there's still a lot that we need to get more confidence in, or get the regulators to have more confidence in, before we can get to that animal-free drug. And I think the closest we're going to get to an animal-free drug right now is with that drug repurposing, is that what's already out there, let's not do any more animal testing, let's do efficacy testing on the organoid models. Because it seems that even with this is a rare disease, there's a pressing need, there's gene therapy. We already have data about how gene therapy have been used, and it still goes into mice and monkeys. So we still have that, that safety data that it's very disappointing. So I don't want to stop on disappointing. I want to kind of maybe give us some things to think about. And this is Lulu, the chimp, who is no longer with us, but I love her face, and I feel like she's telling us to keep going. She's telling us that this is what we need to think about and we can do it. So I feel like some of these we've started to do. So I think the prioritizing the funding for non-animal methods. Like I said, the NIH has started to do it. Hopefully the UK is going to make that move. Incentivizing education and training. My organisation is not really as heavily involved in that, but I think there is some progress there. Queen Mary University in London has just opened a master's course, I think, on organ chips, which hopefully is going to train the next generation of people that we need to be able to take these methods further. Data is the big one. So how do we get better access to the human data? How do we get data sharing? How do we get pharma to share? Not just share the drugs that have gone through animals, but share what have they got in their coffers? What have they got from, how long have they been doing drug development? How much animal data have they got? How much of that could we use to stop animals being reused? I know we don't want animal models, but if we can at least make the legacy of those animals dying computational models that mean that animals don't have to be used for those earlier stages, I think it's worth it. I think it's worth trying to get them to share. And it's, you know, I know they don't want to, so it's worth trying to get them to for that reason as well. And then I think more case studies, the more that we can kind of share, what do we know that's worked? And that's kind of, I'm sorry, I've probably bored the life out of you tonight, but that's kind of what I'm trying to get at here is like, where have we got these success stories? Where do we know that things are working? Where do we know that the animals didn't and these kind of organ chips did? And how can we share that with each other and just build up our evidence base so that when we're having these kind of contentious discussions, when we're in that conflict with people, or maybe not conflict, but just, you know, passionate discussions, we've got that evidence at our fingertips. So we know that these are the areas where the animals are never going to get any better. And these are the areas where as we build the complexity of our organ chip models and all of that, you know, that is where we're going to get more understanding of human biology. And then there's a lot of work about standardizing and validating. I don't think we need to do any more on that. I think it's not going to be quick, but it's going to happen. So I think I'm kind of tired of the arguments that NAMs needed to be validated, and we know that animals never did, but we've almost lost that. And I don't want to get back into saying that animals should be validated, because I don't want to open up that area of research. So NAMs are going to be validated. Okay, let's go along with it. If that means that we can get global harmonization of non-animal methods and OECD guidance that uses non-animal methods, that's going to be a good thing, I think. So I don't know, let's crash ahead with that, non-animal methods. And then my last point, I had some kind of slides and then I took them out, but I did think I'd bring it up with you all and see what you think about it, is that animal chips. So we talk about human organ chips, we talk about, you know, the human liver chips and everything. And it's something that I'm kind of interested in, is really, could we think about animal chips not as part of a drug development pipeline to lead to human medicine, but for veterinary medicine, for ecotoxicology, for those areas that we know they're going to be problematic for replacing animals. And it's already happening. So there's a rainbow trout gill assay that is accepted by the OECD, which is a global body that regulates the standards for kind of methodology, really, all methods. And they've accepted this rainbow trout gill method, cells. So this is cells from fish that now can be used in place of fish. So I feel like maybe there's a bit of room there for animal chips. My organisation, we tried to do a little bit of work with that. And what we found really interesting was that people aren't looking at it from our perspective. People aren't looking at it from a replacement perspective. So the only way that you can get an animal on a chip is if you kill an animal. You're kind of like, really? There's no biobank of animal tissue or animal cells, or, you know, and that's very limited. So I think if we think this is serious, if we think this is something that we want to do, it's going to need a massive amount of investment, and it's going to really need a lot of thought about how do we get, well, it shouldn't actually, it should be more straightforward, because we should be already sharing this information, these animal cells. We should be making the most out of every animal that's sacrificed. They should be available, but we know we know how we work, you know, we know the world. We're all grown-ups. We know that that's kind of not happening. But I think it's something to bear in mind. And I also, actually, because I feel like I've seen criticism of people that want replacement And that criticism is the people who use animals saying, oh, they haven't thought about veterinary medicine, we're always going to need animals for that. So I kind of think, yeah, no, we don't, actually. If we can find an animal-free pipeline to develop human drugs, we can find an animal-free pipeline to develop animal drugs using exactly the same methodology, but just plonking an animal cell in there. So, and then this is Teddy the beagle. Teddy was rescued from pesticide testing. So he was being used in a lab in Michigan for a 90-day pesticide test. And this with 32 other dogs. And my organization got in touch with the pesticide company. It was Corteva who were doing the test, and kind of asked them why they were doing it. Why do you need to do this 90-day test? And they said it was for Brazil. The Brazil regulations required them to have these data. No, maybe it was 1 year data, because 90 days still required. Anyway, pesticide data required by Brazil. My organization is global. There's a wonderful woman that we work with that's one of our colleagues in Brazil, who pretty much went to the Brazilian regulators and kind of said, why do you need these dog data? Why do you need these pesticide data? This compound is already regulated in various other countries. You don't really need them. And they agreed to stop the test, and Teddy got out. And that's it's good for Teddy, isn't it? It's wonderful to see a beagle's ears, a beagle with a tennis ball in his mouth. That's exactly how a beagle should be. But I appreciate we can't do it test by test, and we can't do it compound by compound. So I like to finish on Teddy to remind me why I need to get up in the morning and do it all over again, and for you as well. So thank you for all of what you're doing. And that is the end. So thank you for your attention. I haven't looked at anything in the chat, so I'm sorry if you've put questions, but I can look at that now, or we can, I don't know, you can speak. I'm sure you can speak.
Thank you, Lindsay. That's brilliant. So if anybody else have questions, you want to put your hand up, Emmy will make you available, I believe.
Yeah, shall I read through some of these? Thank you, sir. Oh, look, Pam, someone's waving a hand. I think it was Pam. Yeah, Pam's waving a hand. Go for it. You might need to unmute yourself. Do you want to speak?
To go back to the thing about validation, and we're using animal research and tests as like a gold standard, but we already know that they don't translate to human. So what is the problem when we've got organ on a chip with 87% success? What is the problem with validation? I can't get my head around this thing that what is the hold up here? We already know it's working.
I know.
So I don't understand what is the hold up?
Yeah.
What are we waiting for?
I guess I'm not a regulator, but I think it's the risk averse nature of the regulators having to make decisions about what might be safe and what might be unsafe. And they want to know that if, I take your point, I'm absolutely in agreement with you, Pam, but I think from their perspective, they want to know that if that organ chip was run in my lab and your lab and Alan's lab and Imi's lab, that we'd all get the same results. And until they're confident that that is going to happen, they're just kind of dragging their heels a bit. And I know it's ridiculous because the animals are not the same, and they're more than happy to sit back and go, yes, we'll take the animal data, but that's how it is. So I feel like if that's what they want, that's what the community has got to give them. That's what the scientific community has got to give them.
And really, sorry, it's just like more showing of the data. Is that one of the problems?
Yeah, it's sharing the data, and it's sharing the data so that we can really exploit artificial intelligence more effectively. So it's not just like I don't really understand it, but it's having it as machine readable so that everybody's data looks the same and everybody's using the same words. Because even comparing organ chip results to animals, you can imagine that an animal might have, I don't know, an animal might have a blister on his paw, but how do you do that in an organ chip language if you've got a skin model in an organ chip? We need to use the same words for everything. And that's a real problem, I think, that people just use different terminology that makes it difficult to make those machine learning models. So there's lots to do with that, but I don't see that it should take us long. I think the first step is getting people to agree to share the data, and then I think we can do it.
I just come up with something this week that there could be a problem with the regulators in the UK, that some of the regulators for ASPA aren't trained in up-to-date organ on a chip. So when they're looking at the 3 Rs for the replacement, I think the ASPA says that if there's a NAM available, it should be used instead of the animal. But it's coming to light that the regulators aren't trained in the NAMs, something another group is looking into. But that could be a problem as well, that they're just not using these things. You know, they're there. The NAMs are there and ready, but you know.
Well, I think we've commented before on the training that the regulators provide to their inspectors, because there was a couple of it's hardly exhaustive what they're asked to look at. You know, they're kind of you have to show that there isn't a validated alternative, and it really felt like the researchers could just go, yeah, I've looked really hard, yeah, there's not, I have to use the animals because it's just not there, you know. So we've tried to comment on that this is not good enough. This is really not good enough. And it's, I guess it's impossible for the inspectors to be up to date with everything, so let us help. I'll review the projects, and I can point, you know.
Not enough, is it? You know, you know, experts can help. So
it's
just very frustrating to me. At my level of biology, it's just common sense. If you've got something that's there, then you know, use it, kind of thing. You can't be any worse than an animal, can you? Where you don't know what's going to happen.
Valid point.
You might as well use a thing that looks safe.
Valid points.
Long enough, I could talk forever.
Sorry, Nikki's got her hand up. Do you want to say something, Nikki?
Thanks, Alan. Just thank you very much for organising, Alan and Amy, and Lindsay. Thank you for coming. It might be a very daft question. Just with the liver on the chip, for example, I'm just curious about how does it show an adverse drug reaction? Is it through data? What does that look like?
I guess it's through data. I guess it's that's a really good question, and I think it depends on the composition of your liver chip, and I think it depends on really what you want to look for. So I think you can look for something really as kind of blunt as, do the cells die? So you can look for that viability. So you look for that kind of overall toxicity, or you can look for things much more subtle that you might look for in an animal. So you could look for liver enzymes. So you can see whether or not those liver enzymes increase. So, you know, even the liver enzymes that you might get in a doctor's test, you know, like a human doctor's test for your ALT's gone up, they can look for that as well. So they can look for subtle signs of damage, or absolute, is everything dead and blown apart. So they're pretty sophisticated, and you can match those. I think the advantage is you can match those clinical endpoints to human clinical endpoints because it's a human model.
That's fabulous. Thank you very much.
So there's a question from Deborah. Shall I read it out, Deborah? Do you want to speak? Yeah. So it says, do you think there's a real risk that the animal research community is determined to absorb new technologies into existing animal-based methods so that a hybrid model is the future rather than the full replacement of animals?
That's a good question. I think I'd like to think that maybe there's an element of that, but I'm kind of hopeful, and I might be being naive, but I'm hopeful that the new scientists coming through will not do that. That there's an opportunity with things like that Masters that's just looking at organ chip, with things like there's a course at John Moore's as well that's computational toxicology that I think might be a bachelor's course. I feel like with those sorts of opportunities, if we can start training people from 18 upwards into this is the future, this is the non-animal method, you're never going to touch an animal, I'd like to think that we can move away from that. But I don't know how the animal researchers think really to understand. I think they feel threatened maybe, and I think maybe they feel that there's a compunction, maybe a compulsion for them to use non-animal methods. But I just like to think that we can, I'd like to think we could cost them out of it. I'd like to think we could start charging them for animals' lives, for what they do, you know, so it's not just an easy decision for them to make? I don't know. Sorry, that's probably a really ineffective answer, Deborah. I'm sorry, but I don't think there's anything easy to it. I think I've heard the argument about a hybrid model, and it does come from the animal users, of course it does. But I think we've just got to keep pushing. We've got to keep pushing for more funding for NAMs only, and try and keep that transition moving. I know it's not ideal.
Thank you. Sheila, did you want to unmute yourself and have a speak?
Yeah, thank you. Thanks very much for your talk, Lindsay. It was absolutely brilliant, fascinating.
Thank you.
I wasn't bored at all, for one. So,
God bless you. Thank you.
A couple of questions. One is, how far off are we from having a human on a chip, do you think?
Sorry.
You can answer that first, and then.
Okay. I'm not sure if that's still a direction of travel. I feel like about 5 years ago, the US FDA had a project that was looking at a 10 organ chip model, and I don't know whether or not that project just kind of stalled, and I don't know whether it was because the technology wasn't quite ready. And you know how projects, you get this kind of finite funding period and then maybe it doesn't get renewed. So I feel like that sort of stalled a little bit. And I wonder now whether we've moved on a bit more so that the individual organs now are really, really at an advanced state of development. But I'm not sure that people are really putting them together. And I don't know why that is. That's really interesting, actually, because I hadn't really thought about this. And maybe we've got distracted with validation because it'd be really, really difficult, I think, to validate a human on a chip. So maybe people are just concentrating on their individual pieces. And let's say 10 years, we might start putting it all together, because it's going to be 5 years for our validation.
Right.
But that's a real top of my head sort of.
Thank you. It's interesting.
Yes,
may I ask another question? You were talking about getting the industry, big pharma or pharmaceutical industry, to share data, to have incentives for them, to encourage them to share data, and especially, or not especially, but including data where they failed in testing on animals.
Yes.
How do we, or how would anyone do that? How do you persuade industry to share such data? Because there's so much secrecy, isn't there?
I wish I knew. I really wish I knew. I think we're trying. So in the US, my work's in the US. So we one of our kind of mechanisms of submitting comments to the agency. In the United States, they have user fees. So pharmaceutical companies pay a certain amount to get their drug product reviewed by the agency. So that it's millions. And one of the things that we've put to the agency is, could they reduce that fee if pharma were more willing to share? So is that an incentive? Because we know it's pharma. The incentives are probably likely to be economic. So how can we make it attractive for them? And what our question specifically to the agency was, would they reduce that user fee if pharma were using NAMs? So if pharma submitted non-animal data, how can we incentivize that? So that was our first kind of line, was right, how do we get them to really think about using the non-animal methods? In terms of sharing their data, that's a really difficult one, I think, because that's kind of confidential business information, isn't it? So I don't know how you get that. I don't know. I don't know where you start with that, apart from, I think the EMA, so the European Medicines Agency, has some sort of a safe harbour that might allow data sharing, but that's not going to be for us to see. That's not going to be public. I think if we want to make these bigger machine learning models, I think we're going to have to rely on pharma's good faith. So we might be waiting for a while.
Or maybe they themselves will need to share those data,
maybe,
when eventually they catch up with NAMs.
Yes.
I don't know,
Yeah,
but
and they
it's
are
interesting that you brought that up.
Yeah, and I feel like so as an animal protection organisation, we're not trusted partners for pharma. You know, it's really difficult for us to get to talk to them. But I feel like there's some level of openness with pharma, and for example, the NC3Rs in the UK. You know, the National Centre for the 3Rs. So there are joint projects with which the NC3Rs is kind of acts as sort of a middleman, and pharma will share the data with them for them to do the kind of retrospective analysis of the data. But then that's published as a scientific paper. It's not like the data becomes available in a database. So it's fairly inaccessible for us. But there is, I don't know how we'd expand that sort of mechanism to get what we need, because it's not public for sure. And the incentive there, I guess, is what they get a paper, the publication out of it. I don't know.
I mean, in all other areas of science, we're encouraged to publish data sets, aren't we?
Yep. Yep.
So why not in the case of, you know, people who are testing on animals? But anyway, thank you for answering the question.
So there's one more question in the chat from Daniela. She's also got hands up. So if you want to ask your question, there is also an answer to your question from Nikki in the chat. So just to flag that up for
oh, fantastic, great, thank you so much.
From Nikki's point of view, but Lindsay may have an answer for you as well.
I will reach out to Linda. And Lindsay, thank you so much for the talk. It was very informative. I took a lot of notes, I recorded, and I will consolidate everything together and I'll present it to my MP. But my question is, I have just 20 minutes with him. What is the most important a politician or an MP can do? Is it more on a local level where they can promote those studies for universities, or of course amendments, etc.? But what can I make him do to push NAMS forward, or what can I ask him to do? What is the most important I could do and change his mind? I know that I have a lot of information, but I don't want to just change his mind from an emotional level. I also want to change his mind from a more kind of logic level.
Okay. Thank you, Daniela. That's a knotty question, isn't it?
I think it's a big question. Yeah.
I wish there was a simple answer. I wish I could just give you a sentence and that would be it. I think it maybe depends a little bit on his background. Like what would be the kind of the most compelling argument for him?
Yeah.
You know, so I think we can make the argument on every case, frankly. So we can do it on ethics if he's interested in animals. We can do it on.
I don't think so. That's the thing. I don't think.
So there's that. There's the capacity to be saving money and saving time, because not all the time, but the more that we invest, the cheaper these will get. You know, the same as electric and everything else. The more that we invest, the cheaper these technologies will get. They are faster than animals. They are human relevant. So there's that scientific perspective. But I think it's and it's kind of linked back to the question about hybrid models as well. I think that maybe education is a really important one to get him to consider. And maybe it's if we're seeing traction at universities and we're seeing courses change at universities, what about the curriculum? What about our school curriculum? I was horrified when my son did his GCSEs to find that they were told that preclinical testing requires animals. Full stop, that's it. No mention of anything else. So it's maybe not a compelling argument for your MP, but I think it's something to consider. So yeah, I am sorry, Daniela, I don't know. I'm happy to help you if that would be useful, if you can work out, I don't know, a bit more about his background as to what might be most useful for him.
I can put something together. I also don't want to take up too much of your time, and then Linda, I can email her. I hope you can hear me. But yeah, I will put something together, and I take everything on board what you said, and I'm pretty sure that I can come up with compelling, not just with one argument, with many, many arguments.
Yes.
So I'm very confident that I can do that.
Good. Okay. Thank you.
Thank you.
Lovely. Thank you. Well done for doing that, Daniela. I think that might well be all the questions that we've got. I also found it absolutely fascinating. Thank you so much, Lindsay.
Thank you,
and I think maybe end with a comment that was from Pam, that we have to all be abolitionists and really push to stop all use of animals, which I think is a really important point there.
Yeah.
Did anyone else? Oh, Pam's got a hand up. Go for it. And in the meantime, check out the new article on Wired about organ sacs. It's on our social media platforms.
Yes.
Sorry, can I? And again, thanks. It's been absolutely fascinating. I love this because I'm kind of medical based. So, I thought just as a comment, really, just to go back to your human specific organoids and how to take somebody's cells and test a drug that's specific to them. And I've got an example. I've got a friend who has got secondary breast cancer, and she was put on a drug called letrozole, and she had the most horrific reaction to it. She's had really bad asthma. She was backwards and forwards to doctors, consultants, treatment for asthma. Why has this suddenly occurred? It must have cost the NHS thousands. And a consultant she saw the other day said, like, well, yeah, it's the letrozole that's caused this. So my thinking is that if she'd had some kind of specific, they'd got her cell and tested the drug, because she's actually very susceptible. She's had loads of allergic reactions to things. So if they'd have realised that, tested the drug, it could have saved all that, saved all her suffering that she's gone for months, saved the NHS money, and she would have got some treatment that's, now is, would have been more useful. So it's a kind of a no-brainer to bring all this.
It is. Yeah. I think we have to make the point for that economic saving, don't we? And that's another. I guess I don't know if you've heard about digital twins. I kind of don't really understand digital twins because it seems a bit crazy to me. But I feel like, with everybody wears a smartwatch or something now, don't they? So those smartwatches are constantly collecting our data about, not invasively, but that kind of blood pressure maybe and heart rate and all the rest of it. And why aren't we doing more with those data? Why doesn't Apple have to share those data to build these kind of intact models of humans? Because then that's where your friend might have been able to get some support if they'd known that, those baseline information about her. Perhaps, there's got to be a better way of doing it.
Yeah, I mean, to me, it's just a no-brainer, really. There seems to be, I think the frustration for me is that there's a block somewhere. We've got all these things. Yeah, there's always a sound of a block. Well, we always sort of say, well, animals are better. We've always used them, but they don't work. But these things work better, and it's difficult. It's difficult for me to try and grab hold of that and think,
it's difficult for all of us, I think, Pam. I think
most of that is public pressure. Which is what the FEAT campaign's all about. Obviously, is raising public awareness, because the more people are aware of the situation, the more that they will fight for that cause. And once politicians are politicians, once people start saying this is what they want rather than that, the politicians will move that way because it's in their interest of getting voted in again. And that's individual politicians rather than state government level, etc. But I think it's definitely it's public opinion, which is what April's all about. We're running out of time. I would just like to say it's been absolutely wonderful. Just a mass of information there, and all so incredibly relevant. It's nice to know that now people will be going out throughout April and doing the things that they do, but also armed with so much more knowledge than they had before. I'll just ask you one last thing before we go. You were talking about animal-specific organ on chips, and I was thinking back to what you were saying to be human relevant earlier, and maybe AI technology, but taking samples from the individual so that you can study the individual. I mean, that would be exactly the thing what you would be doing in the veterinary thing, I presume. You take your animal to the vet would take skin cell samples, create the
potentially, yep. But also kind of upstream of that, I guess, because we know that veterinary medicine's kind of expensive anyway, isn't it? So I think personalised medicine for our dogs might be out of pocket for a lot of people, but I guess, considering the number of animals that are used for veterinary medicine, you know, the number of mice and rats that would be used for develop medicines for dogs and cats and all the rest of it, and the number of, you know, I don't know, any sort of companion animal, any wild animal even, why don't we have biobanks of those kind of cells, so that if you're any drug that you want to start from scratch in that kind of pipeline, instead of having those live rodents, you've got rodents on chips, so that you do all of that testing with that. I think it's just really thinking about where we've had advances with humans. Why should it stop with humans? Why shouldn't we adapt it to use it for animals, you know? And one of my this is not really kind of animal chips, but one of my colleagues works on alternative proteins and novel foods, and we're really interested in how do we get a path to market for novel foods that are, you know, non-animal based. So, you know, kind of think about those cultured meats and the lab meats. There's a requirement that puts them through animal testing, if you want to get them approved. So how do we kind of adapt that as well? And might there be a role for animal chips there if there's still a need for a 90-day rat test, or can we do that on a chip instead? So I think there's lots of different products where we could see some value in having these animal chips. It's just whether we could get the investment, you know, whether we could get the interest in it. I think it's a bit of a nuanced one, really. So I think, it's easier to message on human relevant biology for human drugs. I think people are more accepting of that. But I really mentioned the animal chips because I'm kind of tired of the animal using community saying that, you know, we don't understand that veterinary medicine uses animals, so we're never going to solve it, you know, because we will, actually.
And on that very high positive note, which I must say, actually, the entire talk has been very positive. I know there's been low points in there, but it's very uplifting. And you can, for me personally, I can always see the end in sight, being an eternal optimist. But it's so much nearer than it ever was, you know.
Absolutely.
And that's only going to accelerate. Well, thank you, Lindsay, for a wonderful talk. Thank you, everybody, for coming and joining in and asking your questions and stuff. That's it for this evening. Thank you very much. And we will see you next Tuesday, where I will be talking to you, getting everybody ready for the FEAT campaign, and hopefully getting everybody a bit excited. Thank you kindly.
Thank you.
Thank you very much. Good night.
Best of luck.